A fish that looks brown in daylight can blaze neon green at night. A salamander can reveal glowing bones through its skin. A platypus can turn blue-green under ultraviolet light. And some birds carry bright markings that humans almost never notice. These animals are biofluorescent. Their bodies absorb one color of light and release it as another. They are not creating light from nothing, and they do not normally glow in total darkness. Instead, they are transforming light that is already there. That distinction separates biofluorescence from one of nature's most famous tricks. A firefly is bioluminescent. So are many deep-sea animals. Chemical reactions inside their bodies produce photons, which means they can shine without an outside lamp. Bioluminescence is more like switching on a tiny biological flashlight. Biofluorescence works more like a highlighter. Shine the right light onto a fluorescent material, and molecules inside it absorb that energy. Almost immediately, they release part of it again. But some energy is lost through molecular motion, so the outgoing light has less energy and a longer wavelength. Invisible ultraviolet light might return as visible blue. Blue light might return as green, yellow, orange, or red. Switch off the incoming light, and the fluorescence stops. This creates a strange problem. Humans can see many of the colors being released, so why are they usually hidden from us? Because fluorescence is often overwhelmed by ordinary reflected light. Under daylight, every surface is bouncing a mixture of wavelengths toward your eyes. A weak fluorescent green can disappear inside that visual noise. Researchers reveal it by controlling both the light entering the animal and the light reaching the observer. They illuminate the animal with ultraviolet or intense blue light. Then they view it through a filter that blocks most of that incoming light while allowing the longer fluorescent wavelengths through. Without the filter, the animal may just look flooded in blue. With it, the blue background vanishes and the hidden colors appear. Underwater, this transformation becomes especially useful. Sunlight begins losing colors as it travels down through the ocean. Red disappears quickly. Orange and yellow follow. In many marine environments, blue penetrates farther, leaving animals surrounded by a world dominated by a narrow range of wavelengths. Then, fluorescence performs a visual reversal. A fish absorbs some of that abundant blue light and converts it into a color that is scarce in the background. Suddenly, green lines can trace its face. Red rings can appear around its eyes. Spots, stripes, fins, and entire bodies can separate from the blue environment. A large survey published in 2014 documented biofluorescence in more than 180 fish species. A broader analysis published in 2025 listed 459 known biofluorescent teleost fishes and found that the trait had evolved repeatedly, with a strong association with coral reefs. This is not one ancient glowing invention inherited by every fluorescent fish. Evolution appears to have reached similar visual effects again and again, using different molecules, tissues, and body patterns. Eels can contain fluorescent proteins linked to compounds related to bilirubin. Some cat sharks produce bright green fluorescence using metabolites in their skin. Other fish are still molecular mysteries. The patterns may matter as much as the chemistry. Two closely related fish species can look almost identical under white light, then display different fluorescent markings under blue light. That raises the possibility of a private identification system. To a predator without the correct visual sensitivity, both fish may remain similarly camouflaged. To another member of the species, the fluorescent pattern may be far more obvious. Some biofluorescent fish also possess yellow filters inside their eyes. These can reduce the dominant blue background and increase the contrast of green fluorescence, rather like the filters used by underwater researchers. That does not prove every glowing stripe is a message. A pattern can exist without being intentionally used. To demonstrate communication, scientists need more than a dramatic photograph. The correct excitation light must exist in the habitat. The animal's eyes must detect the emitted wavelength. The marking should appear where it can be displayed, and behavior should change when the signal is altered. In some fish, fluorescence may help with species recognition, courtship, or camouflage. In others, it may be a side effect of ordinary skin chemistry. The same mystery has now moved on to land. Under blue light, salamanders that appear dark or mottled can erupt in brilliant green. Yellow patches become luminous. Bellies may glow more strongly than backs. Skin secretions can fluoresce, and in some species, bones near the surface become visible. A 2020 survey examined representatives from salamanders, frogs, and caecilians. Every species and life stage tested showed some biofluorescence, usually releasing green light when illuminated with blue. Later work found something even more suggestive. In many frogs, the blue wavelengths that trigger the strongest fluorescence are common around twilight, when the animals are active. The green light they emit also overlaps with the sensitivity of abundant green-sensitive cells in frog eyes. That combination is difficult to ignore. The correct light is available, the emitted color contrasts with a twilight background, and the intended viewer may be especially capable of seeing it. Some fluorescent areas occur on throats, flanks, limbs, and other body parts used during visual displays. A frog waving a foot or exposing its side may be doing more than moving a colored patch. In the frog's sensory world, that patch might brighten as it moves. Even here, the answer is not universal. Green fluorescence may improve communication in some species. Orange emissions might interact differently with vegetation, predators, or the frog's own vision. Some patterns may have no signaling role at all. Birds add another complication, because many birds can see ultraviolet wavelengths that humans cannot. Ultraviolet reflection and biofluorescence are not the same thing. A UV reflective feather simply sends ultraviolet light back. A fluorescent feather absorbs light and releases it at a longer wavelength. Both can change how a bird appears to another bird, but only one involves converting the light. Fluorescence has been documented in parrots, penguins, seabirds, and other groups. Recent research has also found it widely across birds of paradise. Their feathers, exposed skin, bills, and even the inside of the mouth can fluoresce. In males, many of these areas overlap with body parts emphasized during elaborate courtship displays. Bright fluorescent patches can sit beside exceptionally dark feathers, creating strong contrast when the bird bends, opens its mouth, or presents a particular angle. That makes a signaling role plausible. It does not make it certain. Fluorescence might strengthen a display, but it can be difficult to separate from reflected ultraviolet light and ordinary plumage color. The bird sees all of these channels together, not as isolated effects in an experiment. Then there are mammals, where the hidden glow may be widespread and often completely accidental. Platypus fur can fluoresce blue-green under ultraviolet light. Quolls, wombats, flying foxes, rodents, and many other mammals can show fluorescent fur, skin, claws, whiskers, or teeth. One museum-based investigation confirmed fluorescence in 125 species, representing every living mammalian order. That sounds like evidence for a secret mammalian language. It may instead be evidence that biological materials frequently fluoresce. Keratin, which forms hair, claws, and many other structures, can contain fluorescent compounds. Porphyrins and other molecules can accumulate in tissues or secretions. Diet, age, preservation chemicals, and degradation may change the strength of the effect. Museum specimens are extremely useful for discovering patterns, but a glowing preserved pelt does not automatically tell us what a living animal looks like outdoors. For the platypus, the biological function remains unknown. Perhaps fluorescence has an ecological effect. Perhaps it is merely a chemical property of the fur. Without evidence that natural light triggers a meaningful signal and that another animal can perceive it, the glow is an observation, not an explanation. And that may be the most important lesson hidden inside these colors. A fluorescent photograph shows what is physically possible. It does not necessarily show what an animal normally sees. The natural light could be too weak. The glow could be drowned out by reflected color. The viewer might lack the right photoreceptors. A camera can also exaggerate contrast by using powerful lamps, long exposures, and filters. But when the light environment, the animal's eyes, its behavior, and the placement of the fluorescent pattern all fit together, the glow becomes more than a photographic trick. It becomes part of another creature's visual world. A reef that looks blue to us may contain red and green identities written across fish. A dim forest floor may hold salamanders outlined in wavelengths tuned to twilight. A bird that seems colorful already may carry an extra layer of contrast for eyes built differently from ours. These animals are not necessarily hiding their colors. We are simply arriving with the wrong light, the wrong eyes, and no filter.